Okay.
Okay.
So this is the typical life cycle for a parasitic zooplankton. And so it actually lives in the gills of fish. And this research is kind of expanding right now. One, because we really realize the importance of zooplankton dynamics to our ecosystems. And also because we're getting more familiar with different chemical analytical techniques to really monitor this type of stuff. So I want to go over the subclass Copepoda and just two orders that I see quite often in my samples. So Cyclopodia, and then that's an image of it, kind of what it looks like in water, and then Calanodia. So Calanodia is really cool. They have these very long antennae, and so when I'm looking through a microscope, I can usually see the antenna before I see the zooplankton. These are all holioplankton, meaning they are planktonic for their entire life cycle, unless they're parasitic, which it kind of is a bit different because then they're tied to their host, right? So they're not floating in the water. Most eat phytoplankton. Larger Calanodia actually eat other zooplankton. They also can eat bacteria in the water. Benthic Copepodia actually eat detritus and bacteria, and so the abundances of those species and the species composition actually affects your nutrient cycling and your water chemistry. So what I really wanted to emphasize here is that your species abundances, depending on what they eat, can kind of have its own little food web, right? And so we typically like throw zooplankton as like the basis of a food web, but that can be really a lot more complex. So we'll get into that in a few moments. I want to give you a taste of what their life cycle is like. So we have kind of this image here, and everyone can see my mouse, right? Or no? Yeah, okay, cool. So their life cycle takes anywhere between a week or a year, depending on the species. So what they do is they hatch into Napulus larvae, where they just have a head and a tail, and then they go through 5 molts, kind of like a crab does. And then they enter their copepodid larva stage. And so this kind of looks a little bit more like the actual adult. And think of them as just like adding on body parts as they go along. So they have the unsegmented abdomen, 3 pairs of thoracic limbs, and then they go through 5 molts, and they're finally an adult. The really cool thing about zooplankton is a lot of them exhibit diapause, which means that they're actually able to pause their life cycle based on environmental conditions. And so diapause basically is only during the egg stage. Okay, so the eggs, the female releases the eggs that she carries in like these little sacs that you can see here. And they just kind of sit because conditions aren't favorable. What might be an unfavorable condition for a copepod?
Water pollution.
Water pollution, yeah. Other things could be, not enough oxygen, not enough food, wrong temperature conditions, right? And so these eggs will sit in diapause, basically a stagnated state until the conditions are right, and then they'll hatch. And so this makes zooplankton dynamics a lot more complicated because you never really know how many or what kind of reserve you have in diapause state. Any questions there? Cool. So this one is one of my favorites. So it kind of looks like something out of Harry Potter. It's Diphanosoma. These are mainly filter feeders. So they eat chlorophytes and diatoms and pretty large particles, so up to 154 micrometers, which is something that if you guys took like a scanning microscope, like you'd be able to see pretty easily. And what's cool about them is they show some size selection and discretion based on the size of the particle and the makeup of the particle. Why do you think that might be important? If you're able to choose your food and everyone else just has to take what is around them and you can have the most nutritious, what does that do?
Gives you an advantage.
Yeah, gives you a competitive advantage. Exactly. So you're able to, get the food that is best for your system, right? Best for your fitness, and you might impact the food that's left over for the rest of the community. Okay, so if other copepods don't have that size discretion, they don't have that selection, they're not able to potentially get the most nutritious particles for them. They're just going to have to take what's given to them. Make sense? It was a question. Cool. The other thing that's really cool about Diphanosoma is that they display parthenogenesis. Does anyone know what parthenogenesis is? So it is when females just create females, and there is no standing males in the population. And so female Diphanosoma will just create female diploid eggs, and they will turn back into female Diphanosoma. And that is their base state. And so what this means is they really persist in stable environments in that state, right? Because you don't need genetic variation. The model that you're working with works pretty well. But they will actually switch to sexual reproduction based off of environmental stimuli. And so what could be an environmental stimulus that would make you want to increase your genetic variation and not be a carbon copy of your mom?
Disease.
Disease. Ooh, good one. Okay. Temperature, like not having enough food, right? All of a sudden you're not competing as well with other individuals. But I really like the disease aspect. Yes. And so the female Diphanosoma will make male diploid eggs and haploid female eggs. And then the male diploid eggs will hatch into male diploid individuals that will produce haploid sperm. Ooh, Wyatt, you said predation. Another really good one. Yeah. So potentially if they're getting predated on a lot, I could see that being an environmental stimuli. So haploid eggs plus haploid sperm produce these female epithelial eggs. And so these kind of work like a diapause, but with new genetic variation. And so they'll take up a stimulus again when environmental conditions are right, and then hatch into female diploid eggs, and they will move back into parthenogenesis. So thinking about your question and your problem set, what does it signal if you're finding these epithelial eggs in your plankton toes?
That the conditions are right, or there's some condition that's favoring sexual reproduction rather than parthenogenesis.
Yeah, there is a condition that's going on that is suggesting that they should do sexual reproduction instead of parthenogenesis. So yes, you're getting that environmental stimulus. So some sort of condition has pushed them out of that homeostasis state of parthenogenesis into, okay, we need to sexually reproduce. Does that make sense? Cool. And so that picture right there in red is one of the ones that I've seen in my samples.
I have a question.
Yeah.
How long, does it take for them to switch into sexual reproduction?
So they can. My understanding from the species that I know is they reproduce and die, and so they will switch into sexual reproduction in the lifespan of that species. So I can't give you they're going to do it in 3 weeks or 2 months. You get what I'm saying? Because it depends on the species that you're looking at, because their generation time is different.
I just didn't know if it was it took a couple generations before they knew oh, it switched to
No. So it's a plastic response. And so they hold the genes in them to do this the entire time. And then that stimulus happens and they know, right? I'm anthropomorphizing. They are able to turn on or off those genes and switch into sexual reproduction, which I think is really interesting because that means they're holding the capabilities to have a male and a female the entire time. Cool. Any other questions? That was a really good question, Travis. Cool. So with that, Alex is going to move into macroinvertebrates.
Okay, so real quick, does anyone want to give me a definition? What do you think macroinvertebrates are? You want to define that for me?
Insects in the water that are big enough to see.
Yeah, yeah. So often it does refer to insects. They are large enough to see, and the invertebrate portion would refer to, yeah, I like that. Small things, except not too small. So they also lack a backbone. So we are primarily going to talk about aquatic insects or aquatic macroinverts, including insects, but also crayfish would fall into this category as well.
And I do want to make clear, also like snails and clams are
Mhm.
considered macroinverts, even though they're not something like we first think about.
Yeah, yeah, and they're also not something we're going to cover here, just kind of in the interest of time. So I'm just going to cover a few kind of key orders here. I'm not going to ask you to go down to the family or species level, but yeah, the first one we're going to cover is Ephemeroptera. These are your mayflies. So ephemera is Greek, and that means short-lived, ptera, winged. And this name kind of reflects the short lifespan of adult mayfly, which may only live about a day. These species are generally either scrapers or collectors, so they feed on algae or diatoms. That'd be your scrapers. Your collector gatherers would be feeding more on the detritus. So to identify them, you might have only two long tail filaments. You might also have three on some species. They have 3 pairs of legs, each with a single hook. And then the adult stage have these wings that are kind of net veined. You can see that here. And at rest, these wings stick up, and the front pair of wings is much larger than the back pair. You can see here. And then they have this long and thin antenna, or rather abdomen, and then the antenna are these short and bristly antenna. So this would be more of your adult stage, and these would be more of your nymphs. So your mayflies have incomplete metamorphosis or hematabolous metamorphosis, which basically means they just lack pupal stage, and the nymphs look more similar to adults. However, they lack wings, and the nymph and imago stage is going to be aquatic, or the nymph stage is aquatic rather. The nymphs will have little pads where the wings will eventually grow. So obviously they start as eggs. These are deposited directly to the water by females. Sometimes they're dropped, sometimes they fly and just dip into it. The nymphs, so these are your immature aquatic stage of your mayfly, and it'll live several years on the bottom of streams, under rocks, on decaying vegetation, or in the sediment. And this is the stage where they're generally feeding. In the last several years, they're in this phase, molting multiple times and increasing in size each time. The sub-imago stage, this is also a stage where it is still sexually immature, but it begins to leave the water. It has wings now, but it's a really poor flyer. And this stage won't last particularly long, but it is important. It's a poor flyer, so it's particularly vulnerable to fish. And then this will kind of happen all at once. So they'll emerge from the water generally in May, but also sometimes later in the spring or in the early summer, kind of all at once. So it'd be like a big boom. And it contributes significantly both to feeding fish, just like as a large pulse of food during that period. But also it contributes a lot to transferring nitrogen or phosphorus from the aquatic system to terrestrial environments through predation by like birds or other terrestrial organisms. So this subimago stage is rather short, and it'll quickly transition to this imago stage. This is the adult stage where it's sexually mature, and it is a very brief stage, so it'll generally only live about a day. And they don't feed during this stage. So when we're talking about its feeding guild, we're talking more about the nymph stage. But the main function during this adult stage is simply to just reproduce. So next I'm going to talk about Plycoptera. These are your stone flies. So plyco means folded. This picture, it's not folded back, but I wanted to show you the wings for the ID. But when they're at rest, they're generally laying these wings back along their abdomen flat. A few species do lack wings. We won't really be covering them. I just want to make you aware of that. But these species are often predators, so they will feed on on various immature macroinvertebrates, mostly other aquatic insects. So the front wings, this is like kind of what I thought was particularly identifiable and a good thing to tell apart from all three of these orders I'll introduce. Front wings are much narrower, whereas the back wings are much wider. These only have two tail filaments, which are segmented. You can see that a little better maybe in this picture. They have 3 pairs of legs, but these have 2 hooks on the leg instead of 1. They have these long antennae instead of the short bristly ones. And they are somewhat dorsoventrally flattened, so top to bottom. Additionally, these require clean, mainly in the nymph stage, which is the aquatic stage. They require clean, well-oxygenated water to survive, and they're extremely sensitive to pollutants.
Yeah, so if you're getting them straight from the aquatic environment, I'd really focus on those hooks, as well as the segmenting of the tails. That's what I focus on.
Yeah,
from the aquatic environment.
Yeah, so for the nymph stage, the ones that are kind of like bolded out are going to be more important. I think when I did the little quiz, I used adults. Generally, I just find adults easier. But obviously, later on, I think we're going to do a stream survey. So it might be good to know these characteristics because we'll be looking at the nymph stages more because that's what's in the actual stream. So again, these exhibit the incomplete metamorphosis, much like the mayflies. However, they do lack that subimago stage. So the eggs are laid by the female directly into the stream, but in a sort of sac this time. Then it'll live as a nymph. It'll last anywhere, depending on species, from 3 months to 3 years. It'll molt around 12 to 23 times, increasing in size. And again, during this stage, it's very sensitive to pollutants. And then it'll crawl, when it's ready, it'll crawl up out of the water as a mature adult with wings. And it will only survive for a few days to a few weeks, depending on its species. It generally won't feed, and its main purpose is simply to reproduce. Third one, Trichoptera, or your caddisflies. So trichos means hair. That's generally referring to, it has hairs on its body and on its wings as well, kind of like fine silk-like hairs. So caddisfly, that name it got generally, so in the 15th century, caddis was a word for silk or cotton, like cloth. And these have those silky hairs, but they also in the larval or pupal stage build silky sort of covering or cocoon around them. So that's kind of where they got the name caddisfly, that and the hairs on it. These can occupy, depending on species, almost any of the feeding guilds. Generally, they're scrapers, collector-gatherers, or shredders. So they might consume periphyton, which is algae associated with flora and fauna, both aquatic and terrestrial vegetation. So the key identifying characteristics is they have these long hair-like antennae, 2 pairs of wings with this really fine silky hairs on them, which are narrow close to the body and kind of like get wider further away. And you might notice it's a little bit similar to like a moth. They're really closely related to moths and butterflies, which are Lepidoptera. The only real difference, these guys have the aquatic stage, whereas moths and butterflies don't. They have 3 pairs of legs, all of which are kind of up on the upper third of the body in this paired spur on each tibia. So like it has kind of the same bone structures your leg might be referred to, like the femur and the tibia. So as these spurs on the tibia. Kay, did you want to speak
Yeah.
about these cases?
Yeah. So what do you guys think is important about the case? Why do they make the cases?
Protection.
Yeah, protection. So caddisflies create cases from the environment around them. So they take little bits of dirt and leaves and they put them all together to form this case, which really protects this soft body here. So I included this picture actually on the bottom here, where you see like the gold and the pearls, because caddisflies have actually been collected to create like jewelry and art, and people sell it for a lot of money. So if you guys are looking for a side job, but typically they look a lot more like the ones that you see on the left. So kind of like cemented together dirt. We'll get into it in a few moments about their life cycle. But in addition to protection, this actually becomes an integral part of their life cycle and their metamorphosis.
Okay. So these organisms actually exhibit complete metamorphosis. So the pupal stage will look a lot different than the adult stage. Typically, eggs are laid on masses of vegetation, maybe a hard woody structure or rock above the water line, and then they'll drop into the water. In the larval stage, so they generally have kind of a larval stage. They look a little bit like grubs, maybe caterpillars. They'll have like a hard head and a soft body, and they'll build this case for protection or camouflage. There's multiple types, so it might be like a domed case where they can move around, or a fixed case, something that's attached to a structure. And then they go into the pupa phase, which is a lot more like cocoon. They'll build a cocoon. So basically what they'll do is they'll close up this case, the head, and completely conceal themselves in it. And so that soft body I mentioned earlier, what they'll do is the cases allows water to flow through, and they'll kind of undulate their body a little bit just to facilitate that, so that gases can still diffuse through their skin, and they can continue to respire as they develop. And again, during this aquatic stage, mainly this grub-like stage, they are very sensitive to pollutants. And then they'll emerge from that and come out as an adult. They'll crawl to dry land. They'll live about a month feeding on nectar. They'll be sexually mature at this stage. They're not particularly good flyers, and and they're mostly nocturnal at this stage, and they're also attracted to light. So a lot of people, if they're not super familiar with this particular insect, might mistake them for moths. But their main function, again, during this stage is reproduction. Now I'll give it back over to Kay.
So crayfish is not something people get really excited about, but I do. Crayfish here in West Virginia are actually, we have like a lot of different species. And I do work with crayfish basically for biomonitoring for acid mine drainage with Dominion Power Plant. And so what we do is we do crayfish surveys to ensure that Dominion Power Plant is basically holding up the deal that they are not going to release insane amounts of acid mine drainage into the system. When we actually see spikes, we see the carapace kind of disintegrate. It looked kind of graphic. I tried to find an image of it, but I couldn't back in my camera roll. So crayfish eat a wide variety of items, including small fish, decaying organisms, and other macroinvertebrates. When I'm catching them in the stream, we actually just take a minnow trap and we bait it with chicken parts. And then we let those sit, and then I'll come back to sometimes like 60 crayfish just crawling all over these chicken parts. So it's pretty cool to see. As I mentioned before, they can be quite diverse, and there's a variety of ecomorphs. So by ecomorphs, I mean different color variations. And part of that may be because we don't completely understand kind of the diversity in species for crayfish. We don't really spend a lot of money and time genotyping them. But also naturally within a stream, you can see different ecomorphs. And by ecomorphs, I mean like color variation and morphological variation. So they're not always a sign of bad water quality. I know we kind of attribute them to that, or we say that they are really, really tolerant. And it's true that most of our common species are very tolerant of, bad water conditions that would kill off all those other species we just talked about. But there are some more sensitive species. So I just want to make you guys aware of that. Some are purely burrowing, others are not. And so some you'll never see. They spend their entire life cycle underneath the ground. Others have their own little burrows that they kind of live in, and then kind of you'll see them skittling about. And they exist in a variety of substrates, so not just rocky substrate, but a lot of those burrowing ones actually live in more finer sediment. So I want to make you guys aware of two genuses. So Chimbaris is one. So when I am collecting crayfish, these are actually some of my pictures. So these are the things that I look for. So Chimbaris will have this smooth rostrum. So the rostrum is that nose there. Okay, and it kind of just goes like this, like the tip of a leaf. And when I show you the next one, you'll see what I mean. And then I look at their gonopods. So their gonopods are their male reproductive organs. And so it's kind of like if you were to draw a crab right now, you would draw it like this, right? So this bottom part is at a 90-degree angle, and it would pinch like that. And that's how you'd imagine it. And so that's how I differentiate Chimbaris from the other genus. And I just wanted to include two different species that I see commonly. So Chimbaris bartoni. I see this one quite often in my samples. And then we have Chimbaris carini rotaris. Sorry. And that's the rock crayfish. So that one actually just appeared in my samples last year, which is really exciting because that means we are beginning to basically mimic the environment that was completely wiped out by acid mine drainage in the stream prior. So we're not sure if it, came in from a side stream or if it was a bait bucket introduction, but it's pretty cool that we're seeing some diversity there. The bottom one is actually a blue crayfish that they discovered in some drainages recently, that if you guys do know where they are, shoot me an email because I'm dying to see them. So
They're at the fish store.
No, I want to see them out in the wild where they belong. And actually that's the Florida blue crayfish. There's different varieties. So Faxionis is the other genus I wanted to make you guys aware of. So when you're looking at the rostrum here, you'll see it has a point, comes out to 2 points, and then goes back down. And so it makes it a little bit clearer on that zoomed up picture that I have. And then if you're looking at their gonopods, the male organ is a lot more slender than compared to the other one. And so it's almost like a tweezer instead of at that 90-degree angle. The other thing I wanted to kind of make evident to you guys is the rusty crayfish. So I don't see this a lot in my samples because they weren't going to introduce an invasive species to an environment that they're trying to help, but it is kind of an issue here. So rusty crayfish, they're invasive and they're basically wiping out a lot of our native species. There's some other issues with them in that they compete with smallmouth bass, young of year. There's some evidence there. And what they basically are, they're more aggressive than our endemic species. And so they'll chase them out of like the best hidey holes and increase predation on the native strain. There's also new evidence of crossbreeding between the rusty crayfish and our endemic species. So not only can like a male rusty crayfish breed with an endemic species female, but they can produce viable offspring that then have morphological characteristics that are a blend. And that offspring can actually backcross, meaning that it can then go on to breed either with another rusty crayfish or with an endemic species. And so this creates some issues when we're thinking about conservation, because it makes it a lot harder for us to identify species based off of their morphological characteristics, especially with crayfish when there's not very many guides or people interested in them to begin with. And so you're kind of struggling to identify to begin. So Faxionis used to be called Oroconnectes, but it was divided into Faxionis and Oroconnectes based off of the fact that some are surface dwelling and some are cave dwelling. And so Oroconnectes is now saved only for those that are cave dwelling, while we use Faxionis for kind of the more surface dwelling species. If you're looking at guides though and research, you'll still see the old lingo used. That's because this was only changed in 2017. So it's a relatively new shift. I include a Maryland guide if people are interested in crayfish type stuff. It still uses the old lingo, but it has really, really good pictures of these very small specific traits. And it's what I use for my own research when identifying crayfish. Okay. Feeding guilds, go for it, Alex.
So I'm just gonna, oh, okay. We're sharing a cursor, so it gets a little wonky sometimes. I'm just gonna go over the feeding guilds. So when I was preparing this, I was, and how I'm most familiar with these is referring to macroinvertebrates. So think of those when you're looking through these. You could probably put fish into these groups as well, but I haven't used them for that. So your shredders, these are the ones that are going to feed on large particles of organic matter up kind of in the headwater streams generally. And that includes like leaves, twigs, and they're going to turn them into smaller organic particles that can be fed upon by your collector gatherers. So your collector gatherers are going to feed on those smaller organic particles kind of on the bottom of the stream. Your scrapers feed on diatoms or algae. They're attached under our surfaces, such as rocks or logs. Your filterers, these are again further downstream, feed on really small organic particles in the water column. They might have a fan-like structure on their body if they're an insect, or they might also build a sort of net to capture these organic particles out of the water. And then so predators, when we're talking about macroinverts, these are just macroinvertebrates that are going to feed on other macroinvertebrates, so other like aquatic insects, generally the like larval stage or the immature stage. So for a healthy stream, you kind of want to have some organisms representing every single one of these feeding guilds. If you're missing one of the guild or maybe a couple of the guilds, that might be a good indicator that there's something wrong with your stream that is in some way impaired. So I'm going to talk about some fish that are common here in West Virginia. So first, I want to introduce the family Cyprinidae. These fish, this family is the largest fish family in the world. There's about 2,000 species, all of which are freshwater, and they're mainly present in the northern hemisphere. So this is an incredibly diverse family of fish. They have only one dorsal fin. They have no spines on any of their fins. I'm sorry about this. I think I moved this and the arrow is now pointing in the wrong direction. This is the anal fin. The pelvic fins up here are in the abdominal position. They have these cycloid scales, which are overlapping like shingles, and they're flexible, and they have growth rings, so they'll kind of grow with the fish. And they do lack teeth in their jaw, but they have these pharyngeal teeth. So you can see here, they're kind of located near the back of the mouth, more in the throat. This is just them pulled out. So the central stone roller, it's pretty common here. It's herbivorous or a grazing minnow. So this is it scrapes algae from rocks or logs using its cartilaginous lower lip. So that's one of the key identifying characteristics of it. Its mouth is also horseshoe shaped. So I'm like going through these as well. And then it has these irregular patches of dark scales along its side. For breeding males only, and this is quite distinctive, it has these bright orange fins with a black line on the dorsal and anal fin. I blew that up here for you. The dorsal fin I moved down here. And the breeding male will also have these tubercles on its head. So these are kind of found in your high gradient streams, so kind of your headwater streams, in riffles and pools, and they prefer sort of gravelly substrate. It's a very tolerant species. It can be found in almost any stream where there's enough food to support it. It doesn't matter if it's kind of impaired or polluted.
So yeah, when we do fish surveys of the stream that I was just talking about that's impacted by acid mine drainage, we find astronomical numbers of stone rollers in that stream because it has amazing algae production, because it does not have like a lot of foliage over it. And so they're abundant food resources, and they get larger than any stone rollers I have ever seen. I'm talking like my palm length stone rollers, which is pretty cool to see such a small minnow kind of get that large. Obviously, it's not a great community, right? So we don't have a lot of diversity, so it's not very healthy. But yeah, so that I think that testaments to how tolerant this species is, especially because conductivity spikes and pH fluctuates highly in this system.
So the black-nosed dace, Rhinichthyes atraculus. Again, this is in the family Cyprinidae. This is one we see a lot in our surveys that we do, our headwater surveys in this state. So the dace name comes from dars, which is Old French meaning to dart. Kind of refers to its swimming style. These feed on small insects and other invertebrates mainly. So it's key. The thing I use to identify it is, so here's the caudal peduncle. It's like this bone right here where the caudal fin comes off. It has this black line that extends all the way past its eye to the tip of its snout. Sometimes it's a pretty clear kind of like reddish band along with it. Has a pretty fusiform body shape, so kind of tapering at both ends. Has one dorsal fin with no spines in it, again, because it is Cyprinidae. Its mouth is subterminal, so not directly down, not directly flat, but kind of coming down at an angle. It has sort of a light belly. It might be white, it might be kind of golden or yellow in a dark back. So there's another dace in this genus, which is the long-nosed dace. So this one's the black-nosed dace. You can notice that the snout just barely extends over the lower jaw. So if you're looking at them, you might get confused. The long-nosed dace, the snout comes way out over the lower jaw. I kind of call it the gonzo fish because it has a really long snout. But this is the black-nosed dace, so it does not. The next family I want to talk about is Salmonidae. So this is pretty popular, especially among anglers. And so the key thing here, they have these adipose fins. There's other species that do have adipose fins, but also the jaw extends past the eye. They have like kind of a single row of sharp teeth, no spines in their fins. And the pelvic fin is again in the abdominal position. So further back rather than in the thoracic position. So brook trout, this is like kind of the main sport fish here in West Virginia. Real simply, you can notice it has these light spots kind of on a dark background. It's going to help you distinguish it from rainbow trout, which we'll see later on. Has this sort of worm-like pattern along its back here. I think they call it vermiculated pattern. The lower fins have this white leading edge. And then, so this would be your palatine arch, and this would be the vomer. You'll notice that there's no teeth on the vomer. So that'll also help you distinguish it from the rainbow trout. And again, because it is Salmonidae from that family, it has an adipose fin and no spines in the fins. Rainbow trout is stocked here in West Virginia. Differences between it and the brook trout has dark spots on a light background. So it's literally like the opposite. The caudal fin, the tail is widely spotted, and it also has spots extending past its eye down to its snout. It does have the teeth on its vomer, as you can see here. And again, it has the adipose fin and no spines in any of its fin. So I'm sorry, I guess I didn't mention. So the difference between the adipose fin and the other fins, these fins are like, they have soft rays and kind of like a membranous connection. So like it's webbed. Whereas the adipose fin is this short kind of fatty fin. It's a little thicker. It's essentially just adipose tissue. There's a lot of theories about what it's used for. A lot of people think it's like kind of navigation, detecting stream current for them.
So there's another really cool trout that we stock here, and that's the gold rush trout.
Mhm.
Has anyone caught that? So they're pretty awesome. They're actually a genetically altered strain that we create here to attract tourists. It's kind of like a golden ticket, and to be excited when you get it. So I actually caught one by hand last summer. It's pretty cool. They're very pale, and they're albino. So just a little fun fact.
How in the world did you catch one by hand? They're hard enough to catch with a pole.
It was in a pool and it wasn't doing too hot. So I don't know if you guys have used the term snakehead or like snake. Yeah. So he wasn't looking great. He wasn't a fat boy. But he had gotten himself in Patterson Creek and locked up in just kind of like a little pool because the water had receded. And so he just let me pick him up. And I put him back in, and there's two other ones actually in that stream that I saw later on in the summer because I continued to go back there for crayfish sampling. And they had gotten themselves kind of stuck up above a dam, and they were just feeding on all the fathead minnows and stone rollers they could ever want in their life. So they got pretty fat by the end of the season when I left.
You found yourself a honey hole.
Okay. So last one I want to talk about, and this is just one I like a lot. Your long-nosed gar. So this they primarily prey upon like small fish, minnows, sunfish, shad. They're ambush predators. I'll go into a little bit more of that later. But they exhibit a lot of like ancestral characteristics. So they have these ganoid scales, these rhomboid shaped ganoid scales, which are kind of like a coat of armor. They're not exactly super flexible, but they're really hard. They're made of an enamel-like substance called ganoin, and they have this abbreviated heterocircle tail. So it's symmetrical. The caudal fin is more or less symmetrical. However, the caudal peduncle will look a little asymmetrical because this vertebral column will go up here to the top. And that kind of helps since their fin position is further down and lower on their body rather than up in the center of their body, like you might see on a more modern teleost fish, that would allow it to kind of maneuver a lot better. This will basically help create a little bit of updraft or to help keep it level in the water column. So they're pretty distinct organism. They have this rostrum, this really long, narrow rostrum full of these tiny villiform teeth. And so for this long nosed gar, if you're trying to compare it to another species, you'll look at the length of this rostrum and it should be 2 times or more the length of the rest of the head. So you can see here, this is clearly about probably more than 2 times this length from here to here. Yeah, so they also have their fins. So they have this dorsal fin is way to the back, and the anal fin is also further back, and they're both very wide. And what that does is just give them more surface area to kind of help give them increased burst speed. So these are your ambush predators. They're just going to kind of hang out. And when they see something they want to eat, they're just going to like use those surfaces near the back to really lunge really quickly at their prey and capture them using their long beak and villiform teeth. So they're pretty distinctive as well. So these are predators, but predators of other fish. And Kay, I think you wanted to start this one off.
Yeah, so have any of you guys learned about the river continuum concept before? Yeah, so a little bit of the history. So this was actually introduced by Robin Van Noot, and when it came out, it was groundbreaking. So he released it at a conference and everyone piled into the room and it was like a eureka moment. But now we kind of see it as fundamental. So it was the first time someone really pointed out and emphasized an entire system is functionally linked. So prior to this, fisheries biologists and freshwater ecologists were described as you study your 100 meters of stream and that's it, and you study it to death. And so when he released this concept and developed this hypothesis with a number of individuals, he began to instill this paradigm of thinking of freshwater ecosystems as intrinsically linked. So what happens upstream affects what happens downstream, and it is a combination of your geomorphological characteristics with your energy dynamics that really shape your entire ecosystem. And so this is what really pushed us to study our watershed as a whole. So is everyone familiar? I'm not sure if everyone's familiar with like the term watershed, but that's basically your catchment basin. And so you have your headwater streams that flow into your second order streams, into your third, into a much larger river system that all drains into the ocean. And so I kind of picked up my coffee cup earlier, and I think it's a great example. If you think about your edge as like your first order stream, right? And your center as kind of like your ocean, everything flows towards the center. So what this created was a hypothesis for us to understand the biological and chemical processes, their relationship to our physical attributes, and how these patterns change. And by looking at a number of systems abstractly, we're able to come up with this concept. And it really relates to energy. So Alex, you want to take it away?
Yeah, so we'll start here in your headwater streams. So these are your high gradient streams. They have a lot of riffles, rapids, and falls, and they're often shaded because they'll be kind of covered with trees. They're narrow, so the trees can kind of grow the canopy over the stream and block the sunlight. So there's not a lot of photosynthesis going on up here. But what is going on is you're dropping a lot of organic matter, so leaves and sticks into the stream. So that's your kind of main energy input. And that means you're going to have a lot of shredders, a lot of organisms, macroinvertebrates, breaking that matter down up there. Yeah,
so
and it's, I'm sorry.
No, it's
it's
okay. So
gonna-
they form the basis of your food web. So the
yes.
basis of your food web is not coming from the system itself, like we might think of necessarily in a lake where we have algae and, you know, periphyton and macroalgae. It's actually coming from your terrestrial inputs. And so that's why if we think about like logging in streams and riparian area in headwaters, this is a huge concern because that is what gives you the energy for the rest of your system.
Yeah. And it's also, you're going to have a larger proportion of groundwater input into the stream. So these waters are going to generally be cooler. So you're going to find more species like trout, which kind of prefer those cool, clear waters, right, of the headwater stream.
So to clarify, you have more groundwater in relation to the size area of your stream, right? So your streams tend to be more narrow. They actually tend to be more flashy, right? Because of that groundwater input. And so your groundwater input tends to be cooler, just to make sure that's clear.
So as we travel downstream, we're now looking at sort of the mid-river reaches. So here the gradient's not quite as steep. So there's fewer rapids and falls. The stream is wider. So the canopy is not covering the whole entire stream, and you have a little bit more photosynthesis going on. So you now are starting to develop some aquatic, rooted aquatic plants, so macrophytes, I guess. And you have more, instead of the shredders, you have more grazers. So you've kind of replaced your shredders with your grazers here. You have about the same proportion of collectors as you did before.
Yeah, so you're getting energy inputs from upstream, right? Because now some of that matter has been broken down by your shredders. And so you have finer particulate matter in the stream. And then your terrestrial inputs still matter, but not as significantly as when you were in those headwater streams, right? Because we're starting to widen, we're starting to develop some photosynthesis within the stream. And so it's almost like a hybrid zone when we enter that area. Also, when you're thinking about your habitat, when you're looking at your headwater streams, we really have like habitat in one dimension, right? So we have pools, then we have riffles, then we have, you know, potentially a fall into another pool, right? And so it's very linear in our habitat. Our micro habitats are not very diverse, but as we begin to widen up, those micro habitats begin to appear. And so we have higher number of species.
Yes. So again, the water is starting to warm up as well due to the sun, but also just more runoff from the land as opposed to just the groundwater.
Yeah, so to be clear, it's a difference in your proportion of groundwater to the actual stream water, right? So like your stream is widening, and so you have more water in your stream. And so the groundwater influences it less, and that continues as we begin to move further downstream.
So down here, towards kind of the mouth of the river, the main channel, you're gonna have a lot more dissolved organic matter influencing your system. So you're gonna have a greater variety of both phytoplankton and zooplankton. your water is going to be a little more turbid, so you're going to have less fish feeding on site. But you'll have some areas maybe off channel where there's little micro habitats where you have, again, some of those aquatic plants, those little aquatic communities. And also because the water is more turbid in the main channel, you won't have as many rooted aquatic macrophytes.
Yeah, so this is where DOM becomes important. And I'm not sure if you guys have been introduced to that, but that's dissolved organic matter. And so that's what your zooplankton are feeding off of, right? And then you finally have nutrients enough that are broken down enough in the water that your phytoplankton can kind of feed and begin to really produce, right? And so you have a stream channel also that's wide enough that they can get enough sunlight. Does that make sense? Okay, and then now terrestrial inputs are thought to be insignificant. And I'll put that in quotes because it'll really depend on what system you're looking at. You shouldn't like quote that as the Bible. And when we're thinking about this river continuum concept, understand that it's a generalization, right? It's an abstract thought. But when we see differences in a stream, right, from this concept, it can be one of two things. This stream could be really unique in the way that it functions, or we could have done something to mess it up. Right. And so suddenly we see a high amount of turbidity in the headwaters. Well, that's not where it's supposed to be. So what's going on? You know, is that natural? Is that not? Is that because we are doing some sort of land use practice that is messing with the system? So keep that in mind.
Okay. So if there's no questions on anything we covered so far, and we can go back. If you move up to first portion of your problem set, here I have a poll for you guys. Just answer like each question as we go through it. So this first one, just identify the macroinvertebrate order pictured below. Does anyone want to just kind of either put in the chat or shout out what characteristics you're looking at or you're seeing here that are giving you this answer?
Silky hairs.
Yeah, you can see lots of silky hairs on both the body and the wings.
With the folded wings.
Yep. Yeah,
Yeah.
its wings are folded. And what about the shape of the wings? I think I mentioned that for this species.
They're slender.
Yeah, they're kind of slender here towards the body and then widen out, right?
Yeah, the hairs are going to be
Yeah.
really important. And then if you're looking in the actual stream itself, look for those hooks and look at those tails.
Yeah, and you guys are seeing the poll, right? There should be a poll. Okay, cool. Got a few people.
Okay, next one.
So again, same question. Obviously a different organism here. So again, same. What characteristics are you seeing here? It's helping you identify this.
What are you looking at?
The two long tails.
Two
Yep.
long tails. What else? Because two long tails won't always do it.
Nope.
2 to 3, and then 3 pairs of legs.
3 pairs
Yep,
of legs
3
make
pairs
it an
of legs.
insect. I'm gonna say that 3 pairs of legs make it an insect. Doesn't make it the species. So if you're looking at that back tail, you see the 2 hooks. Back leg. I'm sorry. You see the 2 hooks. So that's what should do it. Okay. Yeah.
I'm only seeing one.
I see 2. Maybe it's because
I
I have
know,
my
but
glasses
I
on.
think it's a shadow. This is an
Exactly.
animated photo, so I
Yeah,
think
was-
it's a
I
shadow.
think K's off.
Yeah.
I have my glasses on. All right, next one.
Okay. Well, also the way it's holding its wings, maybe at rest.
And you have the large wing and then the small one.
Oh yes, yeah, a small one down here.
Yep. So this image is a little harder to see the tails because
Mhm.
you have the egg mass right there,
Yeah, but you can. Yeah, here they are.
but you can make it out. Yeah. So what are y'all looking at? What are you noticing?
The
What do you,
two,
how you, what is it?
the two sets of wings.
Mm-hmm.
Two sets of wings, the way they're shaped, yeah. What about the
Yeah.
tails? They're segmented. So if you're, that's where a really good image where you can start to see the segmentation. Okay, next one.
Okay, my bad. I thought the picture wasn't going to show up. I'm sorry. So you only have to do this to the family. This is a new species. You only have to do it to the family level.
So what's the first thing you look at? What's it got?
Hook jaw.
Hook
Oh
jaw?
yeah, yep.
Spots.
Yeah, it has spots. Those would be useful if you're trying to break it down to the species level. What about its fins?
As a adipose fin.
Yeah,
Yep.
as an adipose fin.
But interestingly enough, catfish also have adipose fins.
Yes, they do.
So,
Hopefully you're not thinking this is a catfish though.
yeah, but I'm just
Yeah.
as a mentioning thing.
Yes.
Because I
Yeah.
love catfish. They also have an adipose fin.
Yeah. Other species do have adipose fins.
But salmon get the
But do they have
Yeah,
any
most recognition.
spines? Does this look like it has any spines?
No.
Yeah. Good point. Okay, so hopefully everyone's good on that. I kind of pulled out the dorsal fin here so you can see it kind of looks like these are soft rays, right?
all right. Go to the next one. So what subclass does this belong to? And just call out the order if you can remember it. Go back.
Copepoda.
Calinota. Yeah. Oh no, Copepoda is the subclass.
Copepoda?
The
Oh.
Calinota is the order. Yes.
Got you.
Yes. And the way we know that is because of the really, really long antenna. We can see it coming. Okay. And then what genus and species is this crayfish? So what are we looking at first for the genus? Yeah, the genus, sorry. The rostrum, yeah. So what is it?
90-degree angle.
Nope. That's a gonopod, so that's the male organ. So we can't see that in this image. We can only see that if he was flipped over.
Like a leaf tip.
Yeah, smooth leaf tip. So that's gonna be which one then?
Bartoni.
Yeah, exactly. And so this is really good because it's blown up, but you can see like this little divot that I'm trying to highlight there. So these are like the small characteristics that I use to identify different species. And so with that key, if you bring it up, we differentiate them by like that little divot. We differentiate them by the crease here between its head and its carapace. So it's very shallow in this species. And then we also identify them by the uniform, I'm going to call them bumps for now, in their claws. And so they're the most common crayfish that we see. Okay, and that is it.
Does
So-
anyone, I can share the results of the poll if everyone's done. Looks like we have 12 of 14. Okay, there we go. There you go. Let's see. Can you guys all see that?
Do you want to go through the results, Alex? The
Yeah,
answers?
I'm just pulling up my paper. So let's see. Looks like the majority said Trichoptera. One person said Plycoptera. So let's see. I kind of am going towards Trichoptera here as well. And I think, so again, if you remember, Trichoptera is kind of, trichos means hair, right? So you can see all these hairs on the body and the wings, right? So that should be what's giving that away for you. Do you want to, whoever, I guess you don't have to, call yourself out. If you want to discuss it or anything, we can. This next one, and I guess I apologize. It really does look like there's 2 hooks down there.
I told
but-
you, I'm always right.
It does, but it's not supposed to. So this is a Feminoptera. It looks like everyone agreed. So hopefully these wings standing straight up was a dead giveaway for you, along with the long, slender abdomen. Yeah. It has the 2, which is completely okay for mayflies. Sometimes they have the 3rd. This one does not. It looks like everyone agreed on that one, which is cool. Let's see. That leaves 1 left. We had some people disagreed. We had more disagreement on this one. So yeah, I think Kay pointed out these segmented tail filaments, right? That's a pretty good giveaway. Also, the real wide hind wings and the narrow front wings. So I'd put that as Plecoptera. Yeah, and I didn't try to trick you guys. I didn't try to put any twice. Maybe I should have. I don't know.
St. Patrick's Day. It's a St. Patrick's Day treat.
Yeah. Looks like everyone got these 2. Yeah, these are your Salmonidae. Seems like that was pretty easy for you guys. And then this is your Cyprinidae. Yeah.
Okay, and then we have, so what subclass does this belong to? So it is Copepoda. Cydidae is actually related to Diphanosoma. So that's the class above Diphanosoma. And then you have the last answer, which I kind of walked you guys through. So that's awesome. So if the research that I'm doing with crayfish sounds exciting to you guys, I'm going to actually have a tech position open this summer. So you won't just be doing that. It's open to juniors or anyone who's going to be here, the year after this, because I know some of you guys are seniors. And so basically what we'll be doing is crayfish surveys and then larval fish surveys. So we'll leave here around 5 p.m. We'll be out for 14 hours straight, hitting like 6 sites up by Mount Storm. You got to be able to kind of hike it in and balance on some really rough rocks, because there's some heavy flows there because it is effluent from like release from a dam. So be prepared for that if you're going to apply. But other than that, it's a pretty fun time. We eat a lot of snacks. It's a long night, but
do we use that for like our internship that we need to do for our major?
You'd have to message your advisor, but I don't see why. I don't see why not. I mean, if your advisor is Kyle Hartman, he probably won't tell you no.
Yep, that's my man right there.
So yeah, that's a good shot.
All right, cool. We'll keep that in mind.
Yeah, in addition to that, you'll be doing habitat sampling for brook trout, which can be a fun time. We kind of just walk in the stream, boots, pants getting wet, doing habitat surveys. Get to see some cool stuff. I saw a bobcat last year, so that's my little shtick for that. So if you guys are interested, just like shoot me an email and get you on the list.
Yeah, and I'll say the crayfish survey is actually a lot of fun.
Yeah.
And Kay loves it when you tell horror stories while you're walking around in the woods.
Thanks. Love it. Yeah, no, that's a lie. Yes,
I'll just start talking about Wendigos and skinwalkers and palemen, like all that good stuff, all that creepy pasta.
Love it. Great. No, I actually love the crayfish surveys because we get a ton of them and you get to see them at various sizes and you get to see them like after they've bred. And so when we're doing spotlight surveys, because we deploy traps, but then we also kind of walk around looking for them with lights. And so you see them like no bigger than your thumbnail to like, across. So it's cool. I don't know. If you want to be a crayfish nerd like me, come join.
Yeah, I enjoyed doing it the times I went out last summer. It is a lot of fun.
So you guys can stay for your discussion questions or you can be free.
Alrighty. Peace. See ya.
Yep, I'm gonna go enjoy this nice weather. Hope you guys get to as well if you can.
Trying.
I think yeah, see ya.
So I'm trying to find the characteristics of the zooplankton in the
Yep.
slideshow, and I'm not really seeing any.
So the characteristic of the zooplankton is that they're heterotrophic and they
And
drift.
then that's it.
Yep.
Oh, okay. Thanks.
It's super simple.
And then when we're done, we just send it into the thing on eCampus.
Yep.
Yeah, there's a
Alright.
link on week 9.
All right. Thank you.
Not a problem, Axel. Enjoy your day.
Thanks, you too.
All right, Alex, if you don't mind